All right, so we're moving on to Chapter 4. Chapter 4 is about eukaryotic cells and the organisms that possess these eukaryotic cells. So we're going to kind of compare and contrast this with our prokaryotic cells that we find within the domain Archaea or the domain Bacteria. So here we're within the domain Eukarya. Okay, so just to refresh our minds that eukaryotic cells have membrane-bound organelles within their cells. So membrane-bound. So they have organelles, which are structures inside the cell that have specific tasks that they accomplish. So we're referring to organelles such as the endoplasmic reticulum, or the Golgi apparatus, or lysosomes, or vacuoles. These are all structures that we find within eukaryotic cells. So they possess membrane-bound organelles, the eukaryotic cells. And we said that all members of domain Eukarya have eukaryotic cells. So under the domain Eukarya are the plants, the animals, the protozoans, and the fungi. So we're going to be talking kind of briefly about each of these different groups with the exception of the plants. I listed the plants here because they are eukaryotic, but they're not really something that we find in microbiology that is infecting humans. So mainly the animals we'll talk about, and the protozoans, which fall under the heading of protists, which are a grouping of single-celled organisms to multicellular algae, fall under that group, and the fungi. So with eukaryotic organisms, we find that some of them, especially the protozoans, have specific structures that enable them to move around. And we saw this with the prokaryotic organisms. So we saw with bacteria that they could have flagella. So there is a similarity that they have these long whip-like extensions that come off of the body of the cell that enable them to be propelled around, enable them to move. But if we actually look at the composition of the flagella and how it's put together, we'll find that eukaryotic flagella is different in its construction from that of prokaryotic flagella. So typically the eukaryotic flagella is much more complex and of course thicker than the bacterial flagella. And of course eukaryotic cells tend to be also much larger in size than prokaryotic cells. Now, besides the flagella, we also can find some other structures that are used for movement, and that is the case of cilia. So cilia are short hair-like structures that typically cover the entire surface. So with flagella, we might have several of them or just one, but when you have cilia, it's a lot, thousands of them usually. So they're kind of like short hairs that are covering the cell, but they're used for movement. So they all kind of beat in unison like hundreds of tiny little oars that are all kind of stroking together to move the little cell through its environment. So once again, mainly we're going to find these in our protozoans, our single-celled protozoans, these structures of cilia and flagella. When we look at a eukaryotic cell, some of these organisms have cell walls, just like we talked about with prokaryotes, that they possess a cell wall. We're going to find with the helminths, which are animals, and the protozoa, which refers to kind of your first animals, right? So zoa always refers to animals. So you think about, you go to the zoo because you're going to look at the animals in the zoo. So protozoan refers to these kind of small, microscopic, single-celled, animal-like organisms. And helminths are worms. So they fall under the category of animals. So these organisms do not have cell walls because animals do not have cell walls around their eukaryotic cells. And the other groups that we talked about that fall under the domain Eukarya are that of the fungi and the rest of the protists, the algae. these do have cell walls. So the structure of the cell wall and what it's made of is important because it enables us to use certain medicines to fight off a fungal infection or an algal infection. All right, so it talks about chitin, which is a type of carbohydrate that is found in the cell walls of fungi. So that is different in chemical composition from when we were talking about the cell walls of bacteria, which mainly had peptidoglycan in its cell wall. And if we're talking about algae, then they have cellulose in their cell wall. So different structural components in the cell wall that we can use different medicines to target these carbohydrates. Okay. So cell walls, once again, provide support and shape for the eukaryotic cell, just like they would in a prokaryotic cell, but the composition of the cell wall is going to be different depending upon whether we're talking about fungi or algae. Now, we said for eukaryotic organisms that they have membrane-bound organelles, and one of the main membrane-bound organelles is that of the nucleus. So the nucleus typically in a eukaryotic cell is one of the larger organelles and can usually easily be seen under the microscope a lot of times. So it's spherical or kind of oval in shape, and it contains all of the cell's DNA. And the nucleus is surrounded by a double membrane that is called the nuclear envelope. So if you ever hear about the envelope, it refers to the membrane that is surrounding it. So the nuclear envelope. And the nuclear envelope kind of resembles in composition the plasma membrane, right? Since they're both types of membranes. The nuclear envelope has tiny channels in it that are called nuclear pores, and that allows the nucleus to be able to communicate with the cytoplasm and for material to be able to move between the nucleus and the cytoplasm. Inside the nucleus, you might find one or more nucleoli. Nucleoli are actually condensed regions of chromosomes where ribosomal RNA is being produced. And the DNA that's found, or the chromosomes we can say that's found inside of a eukaryotic cell, is typically linear, right? So straight molecules compared to the circular strands that we found in prokaryotic cells. So they're going to be linear and they can be condensed into chromosomes, and typically there's more than one of these chromosomes or DNA strands. Okay, so when a eukaryotic cell goes through cell division and it's getting ready to produce new cells, it has to copy all of its genetic information and then divide that information out between the two daughter cells that are formed. And this is typically performed during mitosis or meiosis, depending upon the type of cell that's going to be produced. So these are the processes by which new cells receive copies of the DNA or the chromosomes. Okay, so they're kind of divvied out into the new daughter cells. So binary fission is not something that eukaryotic cells go through, only prokaryotic cells. Another organelle that we find is the ribosome. So this was a structure that we talked about in prokaryotic cells. So they are present in prokaryotes and they're also present in eukaryotes. So the ribosome is responsible for helping to produce proteins. It helps to assemble amino acids together to make a protein. Now, just because prokaryotes and eukaryotes both possess ribosomes, it doesn't mean they're exactly the same though. So eukaryotic ribosomes tend to be larger and denser than those of prokaryotic cells. And so the image that I have here, kind of off to the right, is showing prokaryotic cells, or prokaryotic ribosomes, large subunits and small subunits, and then eukaryotic large subunits and small subunits. And you can see kind of how many bases make up the RNA strands and the different types of proteins that are used to create the large and the small ribosomal subunits of eukaryotes and prokaryotes. So they're not the same. They function the same way though, but the construction of them is different. And so that once again can kind of be key for how we target prokaryotes and eukaryotes with different drugs. All right, so let's talk about some of these different types of eukaryotic cells. So we'll talk about fungi first. And the fungi can be divided out into yeasts and molds. So yeast tend to be microscopic in size. They are not filamentous, but they are unicellular, so just made up of one cell that's typically oval in shape. And one yeast cell can produce up to about 24 daughter cells simply by going through a process that's called budding. And that's what you're seeing there in the image is the original yeast cell, the oval shape there, and off to the left it's producing a bud. And this is a type of asexual reproduction. So only one parent is involved, and that parent is passing all its genetic information on to that bud. All right, so it's producing a new little yeast cell. So colonies of yeast, when they all start to kind of cluster and grow together, and you see this maybe growing on an agar plate, a lot of times they can look like bacteria or a colony of bacteria. So it can be kind of hard to tell the difference between a fungus and a bacterial colony that's growing in culture. Sometimes the fungal ones tend to be a little bit more kind of powdery or fluffier than bacterial colonies. Okay. So moving on to the molds. The body of a mold consists of a long filament of cells that are joined together, and these filaments are called hyphae. And hyphae can get to be very long. They can even be a couple of miles long when they're growing in the soil out in their environment. And hyphae can grow by elongating at the tips, and they keep on adding cells at the tip of it. Now, each part of the hyphae is capable of growth and can even fragment and break off, and the fragment can then start to grow. So this type of fragmentation is a form of asexual reproduction. But there's also reproductive hyphae that usually extend above the body, while the growing hyphae is usually found in the medium. So when we come across a mat of hyphae, it's called a mycelium. So it says the intertwining mass of hyphae. So the hyphae itself is the long thread, and the mycelium is when you've got lots of it kind of growing together. Now, fungi can reproduce by spores. So as it says, these can be dispersed into the air, into the water, into living things. And when a spore finds a suitable environment, it can start to germinate and grow and rebuild the mycelium of a fungus and start to, as it says, produce a new colony. All right, so spores can be produced by asexual or sexual means. So here we're showing the hyphae of a fungus. Like I said, long filamentous threads. And here we're showing kind of these spores, different structures. Usually these are elongated structures that pop up off of the mycelium and get it up into the air where the spores are then released.